Guys, so welcome back. So we're going to continue our lesson about enzymes, so let's just get right into it. So now we're going to go over, ignore these, I don't know why I wrote, now we're going to go over substrate binding, okay, so mechanisms of enzyme activity. So you know a few things, right? So the active site is where your substrate binds, okay? So you can imagine it like each enzyme has a different shape. Every enzyme has a different active site. So this active site would fit this substrate perfectly, okay? Now there's two different models, okay? So lock and key, which is the one I just drew, where they're a perfect fit, okay? Now if we're doing induced fit, what we're gonna say is if we have something like this, and then you have like a square come in here, we're pretty much just gonna say that the enzyme will now pretty much turn into a square. Ish. You know, my drawings are pretty poor, you guys know that by now. But so again, change its shape upon binding. So cofactors versus coenzymes, this again are going to be on your exam. I'll spoil it every full length. So you need to know cofactors, very easy. Inorganic, right? Metal ions. Big one is Mg2 plus. This is going to be used for DNA. And then your coenzymes are organic. Okay, so when you see your FADH, NADH, stuff like that. Both are non-protein helpers, and they're both required for enzyme activity. So these are not proteins, okay? Not proteins. Don't get them mixed up. And then enzyme substrate complex is pretty much like this, right? This would be called the enzyme substrate complex because obviously it's the enzyme plus the substrate. So yeah, so that's pretty much all you need to know. Again, yeah, substrate complex stabilized by hydrogen bonds, ionic interactions, and covalent bonds. Okay, so again, these chapters are obviously very short, but they have a lot of important terms. So definitely write these down somewhere, and we'll get to chapter 2.3. So I'll see you guys there.